Thank you, Manoj. Good morning, everyone. I hope that as we enter this third day of IPC, we can continue the momentum of a strong knowledge sharing and insights. I learned so much yesterday, and I'm excited for today. So I am Lourena. I am a plant pathologist. So today's session is to talk about fungal and bacterial diseases and a little bit the relation with their host. So my presentation is a little bit more holistic. I focus a lot on the pathogen, but this is just to show how important it is to understand the pathogen diversity and the disease dynamics when you want to use your host as a control or any other disease strategy management. Also, just a small disclaimer, I am a Portuguese-speaking native, so you will see a little bit different pronunciation, mainly on scientific names, because I use Latin, so I'll read a little bit more Portuguese than English, because I cannot switch. It's very hard for scientific names. And probably we are right, because Portuguese is a Latin language. So with that, let's begin talking why anthracnose is a global threat. So probably many places where anthracnose, where pepper is grown, mainly capsicum annuum, that is very susceptible to anthracnose, you will find anthracnose. So everywhere. So I just asked colleagues from Nigeria, that is in the Atlantic Ocean. We are in Taiwan, and I even could find anthracnose in Canada, that is a little bit colder, because we know that fungal diseases, mainly anthracnose, likes warm, humid environments. So you will see in the 35 north, 35 south of the equator. That's the risk area, but it can still occur where the host or the environment makes appropriate. They have seen reports of more than 100% in the field losses due to anthracnose, but also some areas can escape the disease, but is still a very, very important disease on pepper, mainly because it's both pre-harvest, so it can attack stems, flowers, and is post-harvest. Most of you are seeing more the problem in your fruit, rot, but is actually a pre and post-harvest. So when we are thinking about the control, even when we are thinking about the host resistance, we need to have this both perspective, not just looking at the fruit itself. So usually you just say anthracnose. And I get excited when someone says, what species? I'm like, yes. So you are talking about one disease, but it's actually multiple species, different species. Just on chili pepper, we have recorded, that is a report of at least 31. I put here 28, that's a safe, but at least 31 species just causing anthracnose. And this has implication in their disease epidemiologies because they act different. Some prefer soil reservoirs for inoculum. Some prefer debris, plant debris. Some prefer their seed transmitting. So we will be talking a little bit about that. So it's one disease, but actually we have a complex of species. And in this tree, you will notice that there are three main complexes that host most of our chili pathogens. So is the acutatum complex. So in the acutatum, you will see one of the major emerging, but also very, very aggressive pathogen, that is Colletotrium scoveli. And also you will find the acutatum itself. So we have the gosporioid. So the gosporioid is another complex that hosts a lot of the pepper pathogens. So here we have the gosporioid, and you have truncantum and bonensis as well. So and this can vary, and I'll show you next a very cool map, but this can vary in the region, but for example, for my friends in Asia, basically you have all of them. So your breeding programs has to be very holistic. You cannot just go one species. This is the most common. You have to sometimes screen at least 3 to 4 species, and I'll show you a little bit a cool study we did in Vietnam. So this is a global pathogen diversity. This is a little bit old. We have to update the pepper breeding, and our pathology team is working now a little bit in Africa. We have a cool project in Nigeria, but we need to fill more information in Africa. But most common there is truncatum here. This is the spore in, no, yeah. This is the spore in green. Very common there, but definitely there are more species. One species that's very common in South America, but also we have identified in Taiwan, is bonensis, but most commonly in Brazil. But the rest of the world is still quiet of that. So if you have a breeding program, maybe you can focus on other 5 instead of 8. And we have acotatum. Acotatum is everywhere in Africa, Europe, in Asia, Europe, Australia, New Zealand. In Americas, it's a little bit less important, but you can still have reports in North America. In the US, we have reports. But in Central and South America, not as much. But it's common. But I'm talking about causal agent. And I'll show you a little bit later what I mean by the causal agent. The one that you take samples over and over, it comes again and again. So the most abundant is the most important, but also because we want a durable resistance, we need to also understand what is the diversity in our region. Okay. And when I talk about this species, I'm not, I have to make a disclaimer, and from now on, everybody in this room needs to use multi-loci. So we all can be able to exchange knowledge and work on the same level. Because before we use morphology, we all know that morphology is, you can. The spores, just the picture, I put color just so you can try to see the difference, but if you're in the microscope for the first time without the others, they look the same. It's very difficult. They are very cryptic species, very close related, and very difficult to distinguish if you are not a professional or in the working a lot with Colletotricum. So morphology is not useful for identify what are the most common species in your area. ITS is the barcode. It's just the standard for all the fungi, just to differentiate, different big groups. But multi-locus, at least 4 to 5 important genes, I believe is in this side, 4 to 5 important genes you have to use to actually understand the diversity in your region. So, and in one country, you can find more than at least 4 pathogens in your area. So these are different studies conducted. So this is in China with more than 1,000 samples in Thailand, and we conducted this one in Vietnam in 2004. 2024 is a 4 years project, so we just did the data in 2004, and you will see that it's not only one pathogen. It's not only one species. So if you are breeding 101, it will not work. At least 4 species will be important, and the most important are Truncanto, Scoveli, and Siamensis. So Siamensis is the same with Gosporioid. Not the same, but before people used to think it's Gosporioid, but it's in the same species complex. So you will breed the most important ones. So multiple species can occur in the same region, in the same fruit. They can occur in the same fruit and in the same production system. So your screening or your breeding has to be pathogen informed, not just very, very brief, ask the pathologist or find one paper. You have to try to get this data of what is the diversity in your region, what is in your farmer's field. So in Vietnam, we use FDA cards. We collected the DNA directly from the fruit symptoms. We brought in to Taiwan, and we did the multi-locus to understand what is the diversity. So we find a very cool insight in Vietnam. Almost more than 80% of the samples came as Scoveli. And in the north, we had more homogeneous, most of the samples hit to Scoveli, but in the south, it had a little bit more diversity. It means that if we, for example, were just using Scoveli, we will have a very good resistance in the south, but in the north, maybe it will have a breakdown. So we need to test at least 3 or 4 of the background material we think is appropriate for that region, also with those other pathogens occurring in that region. So that's why species identification matters, because of the disease dynamics. Not just on host, we know different species may attack different background, different resistant varieties, but also we know that it's very associated, mainly in anthracnose, it's very associated with the timing, the fruiting stage. And this means that each fruiting stage, the green fruiting stage, the mature red, they will show different genes interacting with your pathogen. And also the diversity of species you have in the field. So in the green stage, you have, for example, more cosporioid complex and also acotatum complex. They have been shown to be highly, highly, highly aggressive to green stage. Green stage, yeah. So it's contrary on my thing. And for example, truncatum is more associated to the red stage. So also, if in your market, for example, you are breeding for green fruit, you know what to expect, which ones like the green fruit, and also for red, you know what to expect, and you can adjust your breeding program, but also your management strategy based on that. The disease cycle, I'm a plant pathology, I have to show you the disease cycle, but also it's very important because also these different pathogens, they prefer different ways of spreading, of continuing their life cycle. For example, cocoids, he lives on weeds and they are symptomatic. You don't see the symptoms on the weeds. So if you know that, okay, this pathogen can survive on my weeds, you know what management strategy you need to use. As a plant pathologist, we always want to share for all our partners that although maybe you are more focused on host resistant, you can have a better success if you integrate them, if you already are breeding for a system, if you understand what is the common management your farmers will use. Because I really love the presentation yesterday that even for the harvest, you have to breed for the perfect machine to be able to get the harvest. So it's the same with management. If you already have your background gene of resistance, your QTL, all of them, you need to know, okay, what is my production system? Is in Africa, is very different the way they do in Asia. So you need to understand and already put that as you work. So starting, and of course, host resistant for us is still the first and the most sustainable. So it's our first one. We want to start with a host that is resistant. And there are a few studies, very cool studies, that has been done, identify some QTL on capsicum, and those actually wild veg sources. So, but again, it depends on the fruit stage. It depends on, and is everywhere, as you can see, is in all the chromosomes. So it's very, very hard to come up with a just 101 species, 101 maybe chromosome or gene. So it requires a holistic breeding, mainly for Cholecalciferum. So in terms of host resistant, we need to develop a broad spectrum resistant. We need to understand the link with green and mature fruit. And of course, we need to now use molecular to fast, do precise breeding. So this is a courtesy PPT from Derek. So they already screened the whole call collection of peppers that we have, and you see here they consider less than 4 millimeters resistance. So with this material, they are doing more work on that. And this is just a paper that came up this month from Swarup, where in all the samples they have at least about 2% highly resistant. So this is a material that we can start working to find solutions for anthracnose. Oh, sorry. Next. Yeah, so regarding wild veg, for I think our breeding program has about 20, 30 years, I will say. 20? No, do the math. 40 years. Oh, okay, 40 years. Okay, so I just put some timeline of the past 12 years or so. So we have been working in the region with partners in Asia, in Thailand, Australia, and others, and we have identified resistant lines. We have worked in the transfer of those in maybe from the resistant background to Annam. And Derek team have worked a lot in the segregation population. So now I wanted to team up with Derek, and we have been discussing how we can start looking in more holistic and looking at the population structure of this anthracnose we are seeing in the region and associate them. So we wanted to propose for anybody who want to work with us in this project, where we will do our pathogen surveillance. You send your FTA card, we identify the pathogen diversity, and then we're going to link this population structure with the phenotype. So we will screen in our greenhouse and also in field with multiple pathogens from the region where you are, and see if we can come up with a practical breeding solution that is more durable. Also, just very quickly, I want to share, because most of our partners ask us, how you screen, how is the scoring, how? So every 2 years we provide a training, and this year we're going to have a training, and we're going to do it in Thailand. It's going to be in June. This QR code is, if you're interested, you can scan, later you can fill, and I will send you the information when we have everything ready. So we're not going to only talk about anthracnose. People who know me probably know me more for bacteria wilt. So we're going to be covering most of the important diseases for pepper, tomato, and cucurbits. And APSA members have always discount, and also government institutions. In terms of biological control, I also focus a lot in biological control. We have been looking at other fungi, bacteria, or even plant extract that may be combined with host resistant. So we have 2 candidates in our in our lab. One is a yeast and another is trichoderma that is working very well. So what we did, we wanted to check how much individual compared to if we associate with the host resistant we have currently in the market in Taiwan. So you can see that the yeast show 84% disease severity reduction. So we thought this is a very good example of how you can help your farmer to reduce a lot of the damage on the fruit if you combine both. I'm not going to talk about much because my time is almost done. So I just wanted to share that for future direction with what we need to focus more when we are thinking about the management, definitely we need to do a pathogen-informed breeding. We need to look at the phenomics, genomics, and look more to exploit those to find precise interaction with our host. Also, we are open for collaboration. We want to work with everybody, and we are very excited to maybe understand more what is happening in the region and understand how we can control. Although it's anthracnose, there's a multiple species out there, and we need to understand all of them to ensure a prolonged solution. So with that, I just want to share a little bit about what we have in mind. I don't know if, yeah. So it is a tool that we are developing in our pathology group. It's for this vegetable disease monitoring. It's not just for anthracnose, but also for other diseases. And basically, I don't know, I need... And basically, we collect the DNA of the pathogen. We go in the level of the pathotypes. We integrate with the environment to see if we can get idea of the diversity in the region. And we hope to collaborate with our breeders to understand what host resistant are associated with those in the region, the biocontrols, what culture methods might work, and chemical. And of course, for special project, those will be specifically yet under the agreement, and later they have to become public after all our partners. But yeah, so far I wanted to invite everybody in ASEAN, so I wanted to start with bacteria wilt. So if you are in Asian and you want to collaborate with me for bacteria wilt to send your samples, we can do the phylotype of those samples, and we can understand what are the samples there. And for 2027, we hope to do this for anthracnose. And yes, so it's important for us to talk about the virulence of the pathogen when we are thinking about that. As you saw in the slides, most of these different species, they have different aggressiveness according to the fruit stage, to the host. So it is important for us to work together to ensure we can beat this holistic, without borders pathogen. So to conclude, yes, anthracnose is without borders, is in every continent. We have more than 28 species, causing more than 90% of the fruit decline. In terms of global perspective, we need to use multi-locus phylogeny and focus on the, let's say, 4 complexes that attack Chile. And the disease dynamic is important. Understanding this pathogen is important because we understand what time they infecting our crops, what fruit stage, what host is more susceptible. And this will be important for us to come up with better strategies for our host resistance, for our culture practice, for our biological controls, and overall reduce the damage of anthracnose. With that, I want to thank our World Veg donors, and my team that work with me. This is a work of all of us. The photos on the side are students already left. One did all the sequence of the species in Vietnam, and the other one did the biocontrol work. And up is the pepper breeding, because we work with them, all the resistant QTLs, and follow-up after that is made with them. With that, obrigado.